Okay. A number of years ago, I was working on a big ticket litigation with lots and lots of attorneys involved. And we were really struggling with one claim. We had no idea what the claim meant. So one of the junior associates present in the room kind of quipped, the poetry of claim drafting. To which a very senior litigator present in the group said, there is no poetry in patents. It's all done by bureaucrats. So now we have a situation where a law is passed, and the massive US bureaucracy, called the FDA, takes 3 years to come up with a scheme, a legislative come regulatory scheme that to kind of, enact to sort of express the requirements of the law, and then they end up calling it a patent dance. So this poetic language used by, not poetically, at least non-conventional, non-prosaic language used by FDA to come up with a situation like that almost implies that something went terribly wrong, and they were struggling for something. So now let's get into a situation of what patents were about. But 200 years ago, when patent laws were first being codified, the word invention had a secondary meaning. It was something that an examiner could look at, feel, touch, play with, smell, do something, like a propeller for an airplane or a pill that cures a disease. You could show something. There was a show and tell thing you can still do with the patent office. And then comes things like software and biologics, which upsets the apple cart. Now, software has been banished to Alice in Wonderland. I'm not going to talk about it, but let's talk about what the problems biologics created for this patent come regulatory regime. First, we just describe what biologics are, because that's the key to why things go wrong here. Because it does upset a lot of people thinking about what this technology really means. On the left, you see a small molecule, a traditional small molecule, popularly known as Lipitor. In that molecule, any chemist looking at it could pretty much identify where each and every atom of that compound is. And almost all of the time, if you properly crystallize it, that compound will have a particular three-dimensional structure. So a generic of that compound will have the same structure, look identical. On the right is a biologic or an antibody, for example, or a protein. Generally, biologics are proteins, or mostly proteins. And you can look at the size of the structure. It's massive, it's complicated, it's three-dimensional. There are things that are folding on each other, folding all over the other place, coming together with another protein, and thousands of times larger molecules. So what does that really mean? So right at the bottom, it's called primary structure. That is a biologic protein, for example. A protein is composed of amino acids, which are listed in 3-letter codes at the bottom. And an average biologic protein goes 100 plus amino acids long. And if you still thinking in terms of small molecule structures, each of these amino acids is a small molecule. So once you put them all together and put it in a nice, proper environment in which these can survive, then you have secondary structures form. Parts of this long string of amino acids will form beta sheet structures, a pretty rigid structure, alpha helix structure, another rigid structure, and in between there will be this amorphous floppy links of amino acids rolling around. If just that happens, will there be any function to this molecule? Answer is no. Then you go to a tertiary structure, and now you have what would be a functional molecule. So everything has to fall into place just the right way. And then the active sites and the rest of the structure fall in a way that it can fit into the target and have a biological activity. On the left, we'll also introduce the quaternary structure, where another protein molecule comes in and multiple proteins get together to form the active moiety. And here is, for example, let's take one of the most complex of a biologic, which is an antibody structure. Now, just on the lower right, the structure, there is an extra thing you might notice. Somewhere in the middle, slightly at the bottom, is carbohydrates. So some other molecules, like carbohydrates, have come in, and they have bound themselves to these proteins to make an active antibody. A very schematic thing of what that complex thing on the right is shown on the upper left. Those dark regions in blue and green are the variable regions which form the binding site. That's what binds the antigen or whatever toxin it's supposed to bind. The light blue, light grays, those provide a structure for it. And those light blue, light gray can have a fair amount of variation as long as they create the right structure to present the binding sites in the right format. You can